In this video, I want to show you how in mitosis you can go from a single parental cell that's diploid, or 2N, and you end up with two diploid daughter cells that are exact replicates of that parental cell. So this is mitosis. And we'll start out with a cell, and I'm going to make an imaginary organism that has 3 chromosome types. So we have 23 chromosome types, but that would be hard to draw. So I'm just going to stick with 3 chromosome types. 1, 2, 3. And so those are the chromosomes that came from this organism's mother. And here's another version of each of the chromosome types. Right, so here's chromosome type 1, here's chromosome type 2, chromosome type 3. And again, this is a diploid organism, so it's got 2 versions of each chromosome type. And so this is a homologous pair. So each of those chromosomes in that homologous pair of chromosome type 1 will have the exact same genes that are represented on each version, but there might be different alleles, so there could be an allele for brown hair on this one and an allele for blonde hair on this one, something like that. Okay, let's move forward and let's look at the same cell after DNA replication. We'll still have 6 chromosomes in total. They've just become replicated, so now each chromosome is made up of 2 sister chromatids. All right, so this is a homologous pair. And those 2 are sister chromatids. Incidentally, these 2 are still considered a homologous pair. But let's just take that out of there. Just wanted to do that to be complete. All right. So now that we've got replicated DNA, we're ready to start mitosis. But let's skip a couple steps and get right to when those. I want that a little bit bigger. This is metaphase. Here's the metaphase plate. And during mitosis at metaphase, each of the chromosomes will line up right on that plate. And so there are both of the chromosome type 1, both of the chromosome type 2, and both of the chromosome type 3 lined up right on the metaphase plate. And so this is important. Right now, the sister chromatids are lined up across from each other at the metaphase plate. What's going to happen next is that as that cell starts to divide into the 2 cells, the sister chromatids are pulled apart. Okay, so there's sister chromatids being pulled apart. Okay, and just to slow this down and do some labeling, all of these guys are chromosome type 1. All these guys, chromosome type 2. All these guys, chromosome type 3. But I want to take those labels back out. Oops, one too far. There we go. Okay. And so those are moving apart. So let's look at what happens sort of at the conclusion here. In the end, we end up with 2 cells that each have a version of chromosome type 1, 2, and 3 from mom, from the mother of this organism. And this organism also got a version of chromosome type 1, 2, and 3 from its father. And so that's what you see here at the end. These are still 2N cells. Both of them are 2N cells because they have 2 versions of each chromosome type for all 3 of the chromosome types that are present in this imaginary organism. Okay, and so these 2 cells that we end up here with at the end should look familiar because they are exact copies of the 2 cells that we started with, right? And so, like, I had those 2 alleles, the brown hair and the blonde hair, so we could put those in. They would still look like this. Okay, so that's mitosis. And so just to be complete here, I want to answer the review questions. What is the ploidy of the parental? Let me zoom out here a little bit so we can see the whole thing. What is the ploidy of the parental cell at the start of mitosis, haploid or diploid? And so we started here with a 1N parental cell, or sorry, I'm sorry. We started here with a 2N parental cell, a diploid parental cell. The next question is, how do chromosomes line up during the metaphase? And so this is metaphase. And so the key there is that I wanted you to note that the sister chromatids are across from each other at metaphase. And the second part of that question says, what actually separates during anaphase in mitosis? And so here during anaphase, we see the sister chromatids splitting. Okay. And last but not least, what is produced at the end of mitosis? 2 exact copies of the parental cell. Parent cell. And this is good because this is mitosis, as we've discussed, is how we just make more cells to grow or replace tissue. So we don't want changes. We want just exact copies of the cells that we already have.